Hello, everybody out there. So my name's Shane Hutson. I'm at Vanderbilt, and I'm going to tell you a little bit about a model we developed with CompuCell about secondary palate fusion and how it gets disrupted. And now this is module 5.3 of the workshop for this week, and it's being live streamed and recorded and distributed on YouTube later. So it is being recorded at the moment. You can see the list of funding agencies down there that have supported this work. The first list there is the ones who have supported CompuCell in general in its development. And the work I'm going to talk about was supported by the EPA, and then ORISE, which is an organization that supported a sabbatical I was able to take and spend the time at EPA to work with them. So my main collaborator at EPA was Thomas Knudsen, who's in their National Center for Computational Toxicology, but then a wide range of other people who were there helped out with this project as well. And as always, when we do an EPA talk, the views expressed in this presentation are those of the authors and don't necessarily reflect the policies or views of the U.S. Environmental Protection Agency. All right, so let's get started with this. I want to give you a little brief overview of what the biological question is we were trying to put together, a sketch of how the model works and show you some of the results from it. And then we'll save time to really go into the code a bit and do a couple of exercises on one or two things that were really important to making this work. So this work has been published in Chemical Research and Toxicology a couple of years ago. Tom Knudsen there, Max Lung, Nancy Baker, Richard Spencer, they're all at EPA. So here's a video of what's going on with the model. We'll talk about this in detail. We'll look at all of it. But the take-home messages of what we're trying to put together here. So it's a cell-based model of palate fusion, right? Palate is the top of the mouth, and it has diffused during early development. It includes biochemical regulation. There's a sketch of the biochemical circuits that are active there at the top. And it recapitulates genetic knockouts. One of the things I really like to do with these models is just to look at what an in silico knockout would be, right? How would the genes link into the behaviors we have in the code? And can we simulate knocking out one of the growth factors or another one? And then it provides some mechanistic insight for developmental toxicity. Cleft palate is one of the results of exposure to things like dioxin and a wide range of other chemicals. And we'll see how the model can help us understand the mechanisms and illustrate the mechanisms of how that toxicity works in this particular instance. Now, as I go through this, if you have questions, by all means, feel free to put something in the chat or just unmute yourself and say question, and I'll try to pay attention and answer those. I want this to be more of a conversation than me just talking at you. Okay? So let's take a look at it then. So what's going on with palate fusion? Sort of what's the basis of the biology behind this? Probably the best picture of this is to look at the top right here, the one that's labeled E13.5, E14, E16. Those are embryonic days in mouse. So originally, there's two shelves of the palate that grow down around the tongue, which is labeled there in T, and the two shelves in P. And then they elevate, they sort of rotate up above the tongue and grow towards one another. And then they meet, and they meet here at this midline and eventually fuse into one continuous tissue. All right. Now, palate fusion is the one we're going to look at here, but the idea of tissue fusion is really widespread in development, right? It happens when your diaphragm closes, when the urethra closes. There's some episodes in Drosophila, that last one, dorsal closure there, the optic cup around the eye, neural tube closure. There's all kinds of examples of where this happens. Now, that neural tube closure in the middle gives you a decent illustration of why this is actually a complicated process to have happen, right? You'd think, well, okay, we got two tissues to grow together. Once they touch, well, it's just one tissue, no problem. But it's not sort of a no problem situation because the cells in the tissue are differentiated, right? There's epithelial cells on the surface, maybe a layer of periderm on top of them. Then there's some mesenchyme below that. And so when these fuse, there's all kind of, when they contact, there's all kind of cells caught in the middle that shouldn't be in the middle of the tissue. And if you really want this tissue to become one coherent tissue, you got to get rid of those cells that are left over there in the middle, right? You got to get rid of the epithelial cells and move them out of the way. And that can be done in a variety of ways. They might migrate, they might undergo apoptosis, they might undergo a differentiation and become mesenchymal cell again, right? Some sort of EMT transition. And all three of those happen in palate. So this is some micrographs of what's going on in the mouse palate. And so here PS is the palate shelves, and they're growing towards one another. You can see the staining differentiates a little bit between the outer epithelium there in brown, and then the mesenchyme, which has nuclei and a little bit more of a blue background there. And so when the two shelves first meet, you've got all this epithelium caught between them, and that has to move away. Some migration takes place. You get these sort of triangles at the top, and some of it gets left behind. You can see like this blue stain in D there in the bottom, some cells that are left behind and have to apoptose or undergo EMT to get out of the way in the end. Another view of that, so you have an idea of the orientation. We're going to look mostly in cross section, so it'll look like the bottom pictures here. But if you looked from sort of the perspective of the tongue looking up towards the top of the head, so you're looking up into the palate, it's initially this very large gap. Then at E13.5, the shelves are starting to grow towards one another. They get a little closer at day 14.5, make contact about day 15. You can still see the seam there between them. And then by day 15.5 to 16, they're a nice continuous sheet. And we're going to view these in the cross section instead of this en face view. But for the simulations, we'll do the cross section. All right. So outline of what I'll do, I'll sketch out the computational model, talk about the cellular biophysics and the biochemical regulation, talk about some of the wild type and in silico knockouts, and look at the toxicants effect, dioxin in this particular case. Then we'll dive into the model code a little bit and do some exercises. So the players, right? You got to have different cell types that get defined for any one of these models. And so here's the players that we have in our palette model. In the sort of the cyan and the light green, we've got two different kinds of mesenchymal cells. You can see they have this kind of very spread out morphology here. That's based on how we set the energies, and I'll show you that in a little bit. And those are interspersed within this sort of pale orange, maybe tangerine color that is the extracellular matrix of the mesenchyme. And now surrounding that is our polarized epithelium, and it's got several layers to it. In the blue are some basal epithelium cells, and there are two different shades of blue here because there's different domains, right? The nasal part of the shelf behaves differently from the medial part of the shelf, behaves differently from the oral part of the shelf. So we had those differentiated so we could do different things with them. And over the top of those basal epithelial cells is a layer of what's called periderm. These are very squamous epithelia that are the very surface of all the embryonic tissues in the mouse. And those we've allowed to polarize to have compartments that are basal in the darker purple and apical in sort of the pinker color there. And then underneath the periderm and the basal epithelium in sort of that dark red is an approximation of the basement membrane, right? So it's the second example where, you know, basement membrane obviously isn't cells, it's extracellular matrix, but we're using CompuCell cells to approximate the behavior of the basement membrane. And we're going to use a bunch of focal point plasticity links to give that some elasticity, and the cells then grow on the top of that. All right. Any questions so far? Okay. So what kind of terms do we have in there, right? A CompuCell model is really, it's a Monte Carlo energy minimization problem. So you have to think about, how do I encode my behaviors in terms of a pseudo Hamiltonian or a pseudo energy? And so we have terms for target area and target perimeter of each cell and target length of some cells, particularly the periderm, where we want them to be really squamous. And obviously we have contact energies between all the cells. And just to give you an example of how the contact energies get set up, because now, I mean, we have a system with lots and lots of cell types here. So how do we think about this matrix? and getting the behaviors we want out of the energies that we set up? Well, first of all, you can see sort of what cells are going to be favorable to be on the outside and what on the inside. So if you look and see what are the contact energies with medium, right? It's really unfavorable for the mesenchyme or for the matrix or for the basal epithelial cells to be in contact with medium. A little less unfavorable for the periderm basal cells, and most favorable for the periderm apical cells, right? So they tend to stay on the surface once we put them there. And if you look in a more detail at a couple of these groupings, like what's going on with the mesenchyme. Well, here, there's an energy of 3 for a mesenchymal cell contacting another mesenchymal cell, an energy of 0 for matrix contacting matrix, and an energy of -1 for a matrix mesenchymal cell contact. So what do you think that -1 actually ends up doing to the behavior of the cells?
It favors more contact.
It favors more contact, favors a lot more contact. And what you end up with is these, I was trying to think of the word a while ago, and now is these sort of ramified structures, right? These like extensions and things like that coming off of those mesenchymal cells, because they're trying to maximize their surface area, their contact area with the matrix as a function of their volume, right? So they get all these projections coming off of them. And that's the idea for how we build those. And the reason we put in 0 for the matrix-matrix contact is because although the matrix is modeled as a number of different cells, right? There's a large number of cells in there. We don't really want there to be any distinction between one matrix cell and the next matrix cell. So the contact energy inside a matrix cell is the same as the contact energy between two matrix cells, right? No distinction there. It's essentially a continuous grouping there. All right, so the other thing is to look at the very surface there. And here, you can think about this in a couple of different ways. Let's look at the periderm itself. So in this case, for the periderm, since it has compartments, we have to think about the contact energy between like apical compartments in two separate cells, and the contact energy between apical and basal in two separate cells, and apical and basal in the same cell. And so the parentheses are the internal, the other one is the external. So for example, for a periderm basal and a periderm apical compartment in contact with one another, if they're in different cells or different clusters, there's a very high energy, 30 associated with it. But if they're in the same, it's only 2, right? It's very favorable. And so that tends to then group these together so they stick to the partner in their cluster. And then it's a little bit more favorable for the epithelium to be in contact with mesenchymal cells and with matrix and with other epi cells. So it groups on the inside here. And then the periderm cells have a little bit higher energy in contacting all of those. And so they go to the outside, except for the contact with medium. All right. But now that's just the base. And I think what I want to stress to you and what I'll show you in the exercises that we do is that I find for complicated models like this, it's often not enough just to set up the energies at the start and set a certain number of cells of each type and let it go, that you have to encode ways that the system continually differentiates new epithelial cells, differentiates new periderm cells, because the cells are going to turn over during the course of the simulation. And so you want, as cells die, to be replaced by additional cells. And so you build all of that in as you go. The biochemical regulatory circuits here, lots of growth factors. All of these things in the white boxes are growth factors that are explicitly modeled. You can look at all the chemical fields if you run the simulation. Some of these have activation steps, right? Like Sonic Hedgehog, we follow this link across directly to the left, activates secretion of BMP2, bone morphogenetic protein 2. Some of them are inhibitory, right? Sonic Hedgehog, in contrast, suppresses the secretion of BMP4. And BMP4 has this auto activation of itself there. So there's lots of regulatory elements within this circuit. For what I'm going to tell you, I'm going to focus on 2 little aspects of it. So one part of that Let me get the, yep. There is this feed forward or mutual activation scheme between sonic hedgehog, SHH, and fibroblast growth factor 10, FGF10. One of them is secreted by the epithelium cells, that's sonic, and it diffuses into the mesenchyme. The mesenchyme secretes FGF10, and that diffuses over and impacts sonic hedgehog secretion from the epithelium. So these two tissues reinforce one another. As more epithelial cells grow and secrete more sonic, then that sonic diffuses and helps mesenchyme cells grow and secrete more FGF10. And that loop continues back and forth between the two. In addition, right, those are linked to proliferation, matrix secretion, and proliferation of the epithelium over here. We also have a mutual inhibition loop over here between epidermal growth factor and transforming growth factor beta 3, TGF beta 3 and EGF. They inhibit one another's secretion. And so when you have that sort of mutual inhibition, it behaves like a switch. One or the other can go high, and it depends on initial conditions and a little bit of luck and randomness involved in terms of which one goes high in individual cells. But it tends to go one way or the other. And then those are linked to behaviors, right? If EGF happens to be the chemical that goes high, well, then it suppresses apoptosis, EMT, and motility of the epithelial cells and enhances their proliferation. On the other hand, if TGF beta is the one that goes high, then it suppresses their proliferation and increases their mobility, their probability of undergoing EMT, and their probability of undergoing apoptosis. So that switch then controls what's happening with the epithelium and how likely it is to either proliferate or to die away and move away. All right. And I put in here Ephren B. We didn't put this in terms of, there's not a field for Ephren B in the model, but one of the things that becomes really important for these developmental systems is you often have juxtacrine signaling, right? It's not that there's a diffusible chemical or morphogen that's spreading out through the space. Those occur, obviously, but some of the regulations are based just on contact, right? Does your neighbor have the right kind of cell surface receptors compared to what kind of cell surface receptors you have? Now, we didn't go to the level of putting the cell surface receptors in, but what we do is have a lot of contact-dependent behaviors. And that's one of the things we'll work through in the exercise is how do you check what kind of cells one cell is in contact with, and then make the behaviors dependent on what kind of contact, what kind of neighbors surround each cell. All right. So I'm going to play this as a movie of it, and here's some stills, and you will see that as this plays, maybe a little jerky over Zoom, so I'll go through the stills first before playing it, that the shelves will grow towards one another, make contact about 1,700 Monte Carlo steps, so that should be the equivalent of about E15 in the mouse. And then the fusion process starts, right? We start to get some migration. We start to get some EMT. The little red cells are mesenchymal cells that differentiated. We have a third cell type just to be able to track which ones used to be epithelial cells so we can see them. And some of the other ones, like this blue here, those cells undergo apoptosis. They just disappear if we follow this through far enough. Okay, so let me play that. I'm just playing at pretty high speed here, but it'll loop over a couple of times. Nope, it doesn't loop, but I can play it again. Now, let me tell you one of the secrets of this model in developing it, and it's part of what I want to hit on the exercises. Suppose we didn't have this fancy epithelium in there. We did a very simple, we just had an epithelial cell type and it had contact energy, so it went to the surface. And we ran the model, right? We had some growth and the shelves contacted one another. If that was the case, if we just had a simple non-polarized epithelial cell type, what happens is fusion is way too easy, right? We actually couldn't find a way to break fusion under those conditions. And there are certainly genetic defects where fusion is impacted and doesn't occur properly. And in the model, it just wasn't possible to break it if all we had was the contact energies. So we had to implement these more specialized and polarized epithelia to get realistic behaviors out of the model. All right, so here's a look at what the chemical fields are. This will play again. You'll see the same cells over here on the left, and then the gradients of each of the different chemical fields we're tracking over here on the right will play through as that goes. And let me scroll back through this one a little bit.
Shane, excuse me, can I ask you a question?
Of course.
How did you create that basal membrane to be so small? Because my experience with CompuCell is everything that is below, I don't know, 30 or 25 in target volume, it basically disappears. How did you manage to make it so small?
Couple of different tricks to that. So that basement membrane is actually composed of lots of 4 lattice site clusters. So we would have an individual cell would essentially be 2 lattice sites. That would be its target volume. And it had an incredibly high lambda volume, right? So it was really a very high penalty to even go down to 1 lattice site or go up to 3, right? So it was pretty much stuck at 2. And then we had focal point plasticity links between a group of 2 and another group of 2, and that became 1 cluster. And then we had focal point plasticity links between a cluster and the next cluster and the next cluster. So it was really a matter of having that really high lambda volume and then having the focal point plasticity links that really structured these very tightly together. And that gave us a favorable enough energy that they would stick around.
That's awesome. Thank you so much for telling the secrets behind it.
You're welcome. That's what this is for, right? That's what this is for.
I have another question over that. So basically, you said you set a very high lambda volume, but then how do they move?
All right, now you're testing my memory. I have to remember how we put this in. How do they move if we had that very high lambda volume? I don't think I have a good answer for you. I know that we had to do this. Remind me, and when we open up the code, we will take a look at that and see if we can figure out what it was. I am blanking on what the answer is right now. Other questions? All right. So I just scrolled back through here to stop the simulation of about midway through. And for example, right here's Sonic Hedgehog that's really high sort of in the epithelial compartments and spreading into the mesenchyme. And here's FGF10 that's high in the mesenchyme and spreading into the epithelia, right? Those had that feed forward or auto-active mutual activation between them. And then down here at the bottom, we have TGF beta and EGF. They're both concentrated in the epithelium. That's where they're secreted and where their action takes place. But you can see that TGF beta is already winning, right? It's a bit ahead of EGF. And if we follow this through a little bit more, it gets higher while EGF gets lower. And so that switch sort of flips to the TGF beta, and that's what lets the medial epithelial seam disappear as this moves forward. All right. I think this just looks at the distribution of these, compares to experiment, and we get pretty good, we'll rotate things, pretty good matchup between what gets exposed on the oral surface, the medial surface, the nasal surface, just looking at each one of these and comparing to what's known about experiments. Now, that's no accident, right? This was basically put in. We thought about the behaviors and the coding to make sure that things were secreted in the right places, from the right kind of cells, that we would get behaviors that look very much like what was in experiments. Yeah, there's the Sonic FGF feedback playing on that one. and there's the TGF beta. You can see it really high around those cells that are left over in the medial seam until they disappear. We did in silico knockouts. I'll show you in the code. The way I like to structure this is to have a dictionary that's present from the very earliest part of the simulation, and it's one main Python file and one steppable file, and it's all set up with if statements and conditional statements throughout, so that you can set that dictionary to say, all right, this gene is true, 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 and then to knock out Sonic Hedgehog is false. And then the same simulation runs just with all those conditionals, now treating Sonic Hedgehog as false. And that way it's all in one organization, and you don't have to have separate steppable or main models when you want to do different in silico knockouts. You can just change that one Boolean parameter and run through it. But this is exactly the kind of thing you see when you knock down any of these. If Sonic Hedgehog or FGF10 knockdown occurs, or BMP2, you get insufficient growth of the shelves, and so they don't really make contact with one another, or if they do, it's limited contact, and you get sort of this very narrow neck between them. There's a few of those that don't really give much of an effect in our model. They're sort of modulatory proteins, modulatory growth factors that do subtle things but don't have huge effects, and they don't have huge effects when you knock them out in the biology either. And then there's others that affect the disintegration of the seam. So if you knock out TGF beta, you get this really robust seam that just never goes away, and that prevents the shelves from fusing. If you knock out EGF, you don't really see much of anything. It still occurs and still fuses. And if you knock out a matrix metalloproteinase, MMP9 in this case, then the basement membrane is too stable and it sticks around. And now I'm remembering, we actually have the way you end up with motility is that the basement membrane is continually dying off and being regenerated. And so mobility is not one cell that's moving around all the time, but one piece of basement membrane disappearing and being replaced by another piece of basement membrane. And in this case, that doesn't happen, right? So we get basement membrane that sticks around for a long, long time. And thinking about linking this to toxicology, I'm going to give you the one example of dioxin here. So dioxin is this big, very hydrophobic molecule. It binds very strongly to the aryl hydrocarbon receptor. And what's known about the biology is that tends to increase, to decrease the TGF beta signaling. It impacts it. We don't really have directional effects on this. And it changes the degree to which the epithelial cells apoptose, proliferate, and differentiate. And so it gives changes in the medial edge degeneration, or that medial seam, and thus altered fusion and cleft palate. So what we did to model that within CompuCell is we layered on top of what we have here, right? And there's not a specific aryl hydrocarbon receptor. There's essentially an effect of the aryl hydrocarbon receptor that's coded in that says, all right, we're going to have the receptor for EGF, which is what's happening in biology for this, go up and down with the specific time constants up and down, specific fold excess, and let that affect this balance between EGF and TGF beta here in this switch, right? So if AHR is present and activated, then it increases the expression of the EGF receptor, and that makes the cells more susceptible to EGF signaling. And it sort of pushes this switch towards EGF being the one to go high. And if it does that, then it suppresses all these behaviors, apoptosis, EMT motility, and the seam doesn't disintegrate then. All right. Now, since this is a mutual inhibition switch, for any of you who have looked at those sort of things before, you can get hysteresis in such models. And depending on the parameters we chose for this particular model, we can either have what we'll call a low hysteresis switch. So there's regions here where TGF beta 3 is high, EGF is low, And so the switch has gone towards TGF. And as we increase the EGF receptor, right, there's a place where it transitions. And it transitions eventually to where EGF is going to win the competition. And it's going to be the one that goes high. And TGF beta is going to go low. And in between, we can get intermediate behaviors. And actually, you can get sort of this hysteresis where if you're going one way, the system is stuck with TGF beta high for a little bit longer. And if you go the other way, you're sort of decreasing EGF receptor. It sticks with EGF high a little bit longer, right? And we can have a high hysteresis model where that effect gets even exaggerated. And this is important because the hysteresis impacts what happens when you have transient exposure, right? What if you just got a dose of dioxin at one particular point, and so EGF receptor went up and came back down? Is it enough to cause cleft palate, right? To prevent that fusion at the midline? Well, it depends on how high it went, how much hysteresis here is in the switch, and how long it lasted. But we'll look at a couple of those examples. So if we just have constant TCDD or constant dioxin, so constant activation of this hydrocarbon receptor, and that means the EGF receptor goes up. Well, in this case, if it goes up just a little bit, right, 15% above what would normally happen, the seam sticks around a little bit longer than normal, but it still is able to fuse, right? Sort of these pearls are left in the seam a little bit, and that's seen in experiments, but the seam is pretty much fused. It's okay. If, on the other hand, we go a little bit higher, right here, just at 1.2, we start to have a larger effect. The seam is more resistant to going away. And if we go even higher and have, not even twofold increase in the receptor, but an 80% increase in the receptor, now we really don't get fusion at all, right? It flips that switch so completely to EGF that we can't get fusion of those shelves. So that's uniform, constant activation of those. We can also do transient activation, and C and D up here at the top sort of show you what the patterns are of those, right? EGF receptor starts at 1, goes up, then comes back down to 1 eventually. So these two transients. And so if we take the high hysteresis switch and we go up to 1.6 volts, so this is D over here, and we do that one, this should prevent fusion at the seam. And I skipped the other one. But if we do the low hysteresis switch, we can even go up higher. We can go up to 1.8. And if we do that transiently, it looks like it's not going to fuse, but then the receptor concentration goes down, the exposure to TCD goes down, the receptor concentration goes down, and it can recover, and the TGF beta part can win the switch. So that's what we developed. And what I want to sort of talk through in the exercises, and we'll look at the code, whatever you want to look at in the code, we'll do that first, but then we'll go through the exercises, is to think about the importance of polarized epithelia and how you could put that into one of your models. So I shared beforehand palette model update and palette model steppables.py. I think it's palette model update steppables. Sorry, I should have got that typed correctly there. You should have access to those and be able to open them up. And there's lots of examples in there, and we'll take a look at them. It's fairly complicated code, so we'll also do simpler examples where we just try to make some polarized epithelia and get their behavior to be a little bit better. All right, so before we do the exercise, let me go to the code. How is this in terms of visibility? Do we need to make this text larger for folks?
Yeah, I would say so.
All right, we'll try this. If it's not enough, let me know, and we will work on that. So actually, people had questions about the basement membrane. So let me look that up. Basement membrane. Yeah, so here was where we nucleate little elements of basement membrane. So we're basically checking to see, well, this starts them out, right? It gives a target volume of 3.5. So it wasn't just 2 points, it's 3.5, and a really high lambda volume, right? To make sure that they don't disappear, and just adds them in. So that's nucleating one of the basement membranes. Let me see if I can find the other part about how their behavior is controlled. Okay, lots of examples of BM showing up there. Yeah, okay, here's the basement membrane behaviors. All right, so this will be a good example for us to go through and look at this. So yeah, the code should be in a shared folder. Was it a Google Drive folder, James? I remember
I'll reshare the link. I'll put it in the chat again.
Okay.
And as I say, I was able to upload it and run it on NanoHub, and it ran first thing.
All right, good. So I'm going to go through this little basement membrane behavior. So this is part, let me just explain where this is. Within the steppables file, there's multiple different steppables that get defined. There's one main one that's sort of the palette model behaviors. And as it goes through, it does this every 10 or 100 Monte Carlo steps, however you decide to choose it. And I've broken this up and tried to label it in terms of what kind of behavior we're modeling at each section. So this is the basement membrane behaviors here. And so what we want to do is secrete new basement membrane if we have an epi cell that's in contact with a mesenchymal cell or matrix. So we don't have basement membrane, we're missing it in some location, and we're going to secrete basement membrane if we have some place where it's missing. So we have a Boolean where we can use basement membrane or not. We'll assume this is true here. We are using it. So it's looking at the cluster base cell and asking what type it is. As it goes over, there's a loop over all the clusters. Sorry, that this is within. And if the base cell in the cluster is one of our epithelial cell types, we have a list of epithelial cell types here. So it's the type is in that list. Then the basement membrane secretion probability is 1. Right, this is a very simple one. We have other cases where the probabilities are more intermediate, but this is deterministic for all intents and purposes. And then, right, so we set it to 1, but then we check, and we set it to 1 to begin with. And if that cell is the one we're checking right now, we check its boundary and we see what kind of cells are in contact with it. And if it's in contact with a mesenchymal cell of any kind or with matrix, then if it's not in contact with one of those, we're going to take the basement membrane secretion probability and drop it to 0, so it won't secrete. But assuming it is in contact with one of those, the basement membrane secretion probability is still 1. We have this step here where we're going to allow it to generate different probabilities depending on how often we're calling the steppable. We'll do some practice with that. For 1 and 0, it doesn't really matter, but we set this up so that we could make these something different besides 0 and 1. And then to make it probabilistic, right, we choose a random number, and if it's less than the probability, then we do this action. We nucleate a basement membrane cell. Okay. And as we do that, we're going through and checking the basement membrane cells themselves every time, right? If the cell we're checking is of type basement membrane and it doesn't have 2 compartments, right? It doesn't have that 2 compartments with a focal point plasticity link between them, then we let it die. We let that basement membrane just be disintegrated, right? Cleaved out. And we also put in this matrix metalloproteinase MMP cleavage of basement membrane as a probabilistic event. We have a probability for that. And so if we choose a random number and it's less than that probability, then we take the basement membrane, which is our cluster base cell here under this if statement, and we just set its target volume cell to 0. So we can cleave the basement membrane by just taking one of its parts and setting its target volume to 0. And if there's an isolated part that doesn't have a match, it's going to die off. And we're continually generating new basement membrane the whole time. And that's how the motility of the basement membrane takes place. Okay. Are there other things people want to take a look at in the code before I go to the epithelial part? Okay. So let me go here. I want to show differentiation behaviors first. This is one of the easier things to do, and it'll be part of one of the exercises. In this case, we're starting out with epithelial cells, and so we don't have to differentiate the epithelial cells themselves. But we do allow the cells to, I guess it's not really, you might think of it as de-differentiation, right? They go back to a mesenchymal cell type. And we're going to have that epithelial to mesenchymal transition occur with some probability under some set of conditions. So let's look at what happens with that. So we start 4-cell and compartment list. So this is a loop over all the clusters that exist, and we're going to look at each one of those. We get the centroid of that cell that we're looking at currently, and we assign it to this variable point. We initially set our epi switch rate to 0.001, 1 in 1,000. Okay, and we're going to build all of these up based on probabilities. We either make the probability a little higher or a little lower, depending on conditions, and then we evaluate what's going to happen based on the probabilities. So we start out with 0 probability for either one of these kinds of transitions, either epithelial to mesenchymal or epithelial to periderm, both 0. But then we go down here and we ask, all right, what is our epithelial cell in contact with? All right, so this is boundary cell is a function that's written into here. You can look up the function and see exactly what it does. That returns true if this cell is in contact with any of these kinds, any of these types of cells. And so this first one is checking, is the cell in contact with medium or one of the periderm types? And if it is, this returns true. And we have if not that. So this proceeds only if it's not in contact with one of those kinds. And then it also asks, well, is that cell in contact with one of our mesenchymal cell types or the matrix? And so it only gets here, right, inside these pair of if statements, if our epithelial cell is in contact with the mesenchyme and is not in contact with the medium or the periderm. So this would be an epithelial cell that in essence is lost, that's somewhere within the mesenchyme and surrounded by mesenchyme. And if so, we want that particular kind of cell to have a high probability of undergoing this transition, becoming a mesenchymal cell of some kind from its initial epithelial cell type. And so what we do, if it gets in here, we add a little base to the probability, so plus equals here. And then we also increase it with an amount that depends on the TGF beta field and an amount that depends on the EGF field, right? And the Hill function is a function that I defined in here. I've talked with James. This is something that we want to put into CompuCell. It's really useful to have these sort of Hill functions around so that we can say, all right, what's the maximum probability or maximum rate we should have out of this? And what's the AC50, right? At what point do we get 50% of this behavior based on this field? And so, you know, a Hill function is just this sigmoidal function that goes up in the middle of it where you get 50% of the maximum is at the AC50. So we increase the probability based on the TGF beta field, and we have this minus equal. We decrease the probability based on the EGF field. So EGF suppresses the transitions, TGF beta increases it. Right? So that's changing our EMT probabilities. Then we have another else here. So if it wasn't under one of these conditions. Let's check some other conditions. So we have a not is boundary cell with one of these mesenchymal cell types. So it's not in contact with the mesenchymal cell type. Then maybe that epithelial cell will become a paraderm cell, right? It's not in contact with the mesenchyme anymore. It's surrounded by other epithelial cells or other paraderm cells. And if it's in contact with the medium, the very outside, then we really increase that transition probability of EPT, epithelial to paraderm transition. Okay? So we've got a base transition, and all of that has set is what's the probability that one cell, that when we visit it, will make a decision to undergo one of these transitions, either to mesenchyme or to paraderm. Now, one of the problems with that is that probability will give you different behaviors depending on how often you call the steppable, right? If the probability was, say, 1 in 100, then if you call the steppable every Monte Carlo step, then after about 100 Monte Carlo steps or so, it's likely that most of the cells would have differentiated. But if you call the steppable every 100 Monte Carlo steps, then you're not going to get complete differentiation until 100 times 100, right? 10,000 Monte Carlo steps. You get very different behaviors depending on how often you want to call it. And I always want to set these up so that you can get the same behavior no matter how often you call the steppable. You can do some test where you only call it every 100 steps and speed up your simulation. And then you can do a production run where you go back to calling it every 1 or 5 or 10 Monte Carlo steps and get the same behavior still. And so that's what these two lines here, I'm going to highlight them, do. It's just some basic probability theory that depending on what the frequency is with which you call the steppable, it changes the probability according to this. Essentially, you have to say, what's the probability of it not differentiating over some number of steps that are in between your calls to the steppable, and then the probability of it differentiating is 1 minus that. Happy to talk about the math more if people want to understand what that is. So we have our probability, and then we test the probabilities, right? Choose a random number. Is it less than the probability? And if so, then we got some strings that print out some information about it. I like to do that. And we simply change its cell type. It becomes a mesenchymal underscore EMT, one that underwent epithelial to mesenchymal transition. On the other hand, right, if we want to check the other one, another random number, is it less than that EPT probability? And it differentiates into one of a different cell type depending on whether it was on the nasal, oral, or medial part of the shell, right? But here's that cell type equals new cell type. Easy enough for differentiation. The complication is all in the probabilities and calculating that. And you could put almost anything you want in there, right? We've got probabilities here that depend on the chemical fields, what kind of cells it's in contact with, and we let all of that information then play into the probabilities for these different kinds of differentiation. Questions?
I think we're doing pretty well. Are
Okay.
people able to run it? I know 1 or 2 people seem to have trouble getting it to run, but it ran perfectly for me first time. I know several people said it ran fine for them. Is there anybody who is not able to run it at this point?
All right.
Yeah. It runs on NanoHub. If you're having any trouble, just let us know.
All right, and so that's within the main steppable file. There is another steppable that's in here that will go down to, I'll search for it with apical, that should find it. It gets through all the other calls to apical. Yes, it's called manage epithelial polarization. And so it was worthwhile to have a separate steppable that took care of this, and it gets called every 10 Monte Carlo steps in the way we've set it up right now. let me scroll down here a little bit to the part that is the main section of it. All right. So if you scroll down to the function check apical domains, so what this is going to do, right? This is really the steppable that manages those polarized paraderm cells that have apical and basal compartments. And what we want to make sure of is that, one, if we find a cluster that only has a basal compartment, that it makes a new apical compartment. So we get a whole cell, right? An appropriately polarized cell. And likewise, if we find an isolated apical compartment, we want to make sure we nucleate a basal compartment so we get that polarized cell back again. And so the first part of this is just checking to see what's going on with those. And there's some important steps here in terms of checking over neighbor lists that I wanted to show you. So let's see, where do we want to start with this? All right, we can skip down here to this part. All right, we're checking over a cell. Sorry, there's a question in the chat about trying to get a link, James, if you could take care of that.
I'll take care of that, yes. That's fine.
So we've checked up here basically to find out whether the cluster has one cell that's an apical type and one cell that's a basal type. I guess it's worth looking at that, right? So for cell in compartment list, so that's going through our list of clusters. We get the cluster ID for that cell, and we check whether that cell type is in what we've called typeset apical. And in this case, that's just a variable that basically has a cell type in it. We're looking for an apical cell type here. And if it exists, then we're going to let the apical cell variable become that cell, right? We're going to reassign those. We also check if it's a basal cell type, and if so, we're going to call basal cell equals that cell. Now we come down here. Let's see. Is there a question? Someone, Lily, raise your hand. Maybe that's left over. Okay. If the apical cell and basal cell exist, right? In CompuCell, this is an easy way to check whether something exists. You just say if that cell. And so if it's true, then that means they exist. Then we're going to come down here and check whether they're neighbors. And so that's this little bit of code right here that's important. And the naming convention in this code right now is not great. So let me be very careful about explaining it. This is just a leftover. One of the updates for version 4.3 of CompuCell changed how this neighbor data is organized. And so my names really don't match up with how it's organized very well. But there's this function self.get cell neighbor data list of a particular cell. And so that will return actually a list of tuples. And the first element of each tuple is just the neighboring cell, is one of the neighboring cells. And the second element of each tuple is the contact area between the cell we're testing and the neighbor we found. All right, so for example, cell 1 is in contact with cells 2, 3, 4, 5. Then neighbor surface data would have cell 2 and the contact between cell 1 and cell 2. Cell 3, the contact area between cell 1 and 3. Cell 4, contact area between cell 1 and 4, etc. And so what we can do is set up the iterator like this, which is a little bit of an oddity in Python, but Python lets you do this. And we say for this pair of things, neighbor surface data, comma area, in this list of tuples. And so the first element in each tuple gets assigned to neighbor surface data. So that's actually the cell that is the neighbor. And the second element of each tuple gets assigned to the area there. And so neighbor surface data, although it's poorly named, that really is a cell. That is a neighboring cell there.
It would be the cell ID in this case, right?
No, I think it's the cell itself.
The cell itself, okay.
Yep.
The
cell.
the objective cell, okay.
Yeah, and again, my apologies for the naming. There was a change in how things are organized, and I didn't have time to change the naming. So then once we're looping over all those neighboring cells, we check if the cell exists and its ID is equal to the basal cell ID. Well, great. We say, all right, the apical and basal domains are in contact with one another. They're neighbors. And so we set that touches basal flag to be true. Otherwise, that touches basal flag was started as false, and so it stays false until it's true for one of these pairs of neighbors that it compares. And if it gets through the whole list of neighbors and never set that to true, then it was still false. And so we get down here at the end, and this flag we can use later on to answer different questions about it, right? And what it actually does is it gets returned here, right? What this check apical domains returns is the touch. Let's see, where's the touch? That's right. That should return that flag value. Let me see if. All right, well, this one doesn't look like it's returning anything at the moment. It's just printing out whether they are in contact with one another or whether they are not. It's just a warning if they aren't in contact with one another. The next function down, this update apical domains, this is the one that actually does the nucleation of new apical or basal domains depending on what's missing. So it's going to loop over all the cells, see if it's in one of the polarized compartments list. So that's our paraderm list. Get the cluster IDs and check all of those to see whether the cell is appropriately polarized, whether it has both an apical and a basal compartment here. If it does, great, it goes on, right? If polarization flag is true, pass. That means it just skips that. We're good to go. But if polarization flag is false, we get to the else part here, and it's going to go through and try to say, all right, I need to nucleate this new cell here. So let's think about this situation for a second, right? So you've got one cell somewhere in your CompuCell domain, your lattice, and you want to give that cell a matching complementary compartment, right? Basal make an apical, make an apical, apical make a basal, vice versa. So it's one thing to say, all right, we need to make a new cell. But then where are you going to put that cell? Right? So what's important is we've got to create the new cell, assign a few lattice sites or pixels to it, and hopefully lattice sites or pixels that are already in contact with its matching compartment, or its complementary compartment. And then we got to set any of its parameters, like its target volume, target surface area, those sorts of things. And that's what all of this code here does. Right? So if it's an apical cell type, if cell type apical, we're going to get the list of pixels on its surface. And there's a function built in here that'll be useful to copy into the exercises, where it will return a list of the pixels in a cell that contact the medium and a list of the surface pixels in a cell that contact something besides medium. that turned out to be what was useful for doing this sort of differentiation here. And so if we're checking an apical and we want to create a basal, we want to create it then on the mesenchymal side of the apical compartment. So we're going to check whether the length of the list of other surface pixels is greater than or equal to 2. We want there to be enough surface pixels that we can create something there. And if it is, then we're going to try to nucleate a basal cell using some of those surface pixels, those other surface pixels. So we get the first point in that list of other surface pixels. And here's the steps. So we make a new cell, as easy as self.newCell there. And we tell it what type we want that new cell to be. It's one of our basal types. Once we've created that new cell, then we assign one of the lattice sites to be of that cell, right? Self.cell field, and that list of X, Y, and Z becomes new cell, right? It's assigned to that one. And then this set of if statements here is just assigning a few more pixels from that boundary to the new cell. So we're going to nucleate it at multiple points along there, give it some initial volume. I found in sort of modifying this for the update that if I tried with just a single pixel or single lattice site belonging to the new cell, that they almost always just disappeared before the next Monte Carlo step. It was impossible to keep them. So you had to nucleate it with multiple lattice sites to begin with. So we've defined this, we've created the new cell, assigned some lattice sites to it, assigned its cluster ID to make sure it matches the apical cell that it came from, because when we created the new cell, it got its own new cluster ID, right? A completely new one. It doesn't know it belongs to this cluster until we do this line here, reassign ID flag. And then down here a little bit further, we set the target volume, target surface, lambda volume, length constraint if we want it for the new cell that we just created. And those are based on how much contact there was between the original apical or basal domain and the surrounding cells. That helps us define what the new target volume and target surface area are. Questions before we get to the exercise? All right. So what I would suggest, right, for this first exercise is open up the simulation called nucleate compartments without a number on the end of it. This nucleate compartments.cc3d. And if we go to the steppable file in that, I wrote the challenge in there. I can also go back to the PowerPoint and share it here. All right. Let's see, let me show you this. Let me go back here. So I'm going to open up this nucleate compartments and open it in Player. Stopped it when it was running. All right, so the current, like that nucleate compartments, all it's doing, it's basically a cell sorting simulation to start with, just to give us a base, right? It gives us this globular cluster of cells to start with, and it randomly assigns half of them to be epithelial cells, type epi in this one. And the contact energies are such that the epithelial cells will tend over time to end up on the surface, right? So you don't have to worry too much about the contact energies as you set this up. But now let's think about what's actually happening. Suppose we want to do a simulation here, and we want there to really be a monolayer of epithelial cells around our cluster here, our cluster, our group of cells. This might get us there eventually, but it's going to take a really long time. And in reality, what we'd like to do is have the cells that are on the surface just differentiate into epithelial cells. So I'm going to stop this and go back to the assignment. So what we want to do is we want to create something that looks like this picture down here at the bottom right. All right. we want our mesenchymal cells all to be in the middle and our epithelial monolayer to be on the outside. And your assignment in the exercise here is to start with that nucleate compartments as it is right now and comment out, and I'll go back in and show you that line that just does random assignments of epi cells. We want to get rid of that. And I want you to put in there, change the steppable so that it checks to see which cells are in contact with the medium. And if a cell of type mesenchymal is in contact with the medium, then differentiate it to type epi with a probability such the outer rim of the blob and only the outer rim becomes epi in about 1,000 Monte Carlo steps. Right? So loop over all the cells, check whether each cell is in contact with medium and whether it's of type mesenchymal. And if a cell of type mesenchymal is in contact with medium, convert it to type epi with an appropriate probability. If you get that really fast, then go back and try to write your steppable using Poisson statistics as James chimed in. So the time required to differentiate the outer rim to epi is the same regardless of whether it's called every 1, 10, 50, 100 Monte Carlo steps. All right, so I will let you get started on that, and please feel free to ask questions if something's not clear as you try to do it. I will show you that the one you should comment out is this: if random less than 0.5 cell type equals epi. We want to get rid of that. So if we do, then currently nothing is of type epi. The only things that are going to become type epi are cells we allow to differentiate into that cell type as it runs. And I guess there is a poll. I'm going to launch the poll about completion of the exercise. I don't expect anyone to be done yet, but it's launched so you can let me know when it is done. Oh, it's already commented out. Okay, good. So James says he's having trouble running the example. One thing different about the example is that it does have an XML file. I completely eliminated the XML file on the Palate model, but this one does have an XML script.
I wonder if there isn't a 1-character typo in the XML. It's odd because people were having trouble running the full model on the newest version of CompuCell, but it runs on NanoHub just fine. But the nucleate compartments one is crashing for me on NanoHub. I don't know if anybody else is able to run it.
In
So
431,
It's
the
crashing
exercise
for
runs
me
for
too.
me.
It's crashing for me too on NanoHub.
It's interesting. I wonder what's different because you'd think that they both either run or not run. But in fact, one of them runs on NanoHub and not local, and one of them runs local and not NanoHub. I'm not sure.
Yes.
Let me see if I can find. I'll take a quick look. But
Okay.
but I think everybody can run everybody can run the full model on NanoHub. And I think how many people are able, maybe just a show of hands, how many people can get the nucleate compartments simulation to run?
I'm running local. I don't know if you asked that, right? You're asking
Well, no,
NanoHub.
that's fine. So no, either way,
Yeah.
no. I was
Okay.
gonna say, if people have a local install, then they can run it local.
Yeah.
And somebody says the example is also crashing on NanoHub. So for some reason, let me look at the code a little bit quickly for nuclear compartments. Why don't you keep going? But let me see if I can see what the code problem could be.
All right. Yeah, sorry about that. I've only tested it on my local installation. I've not tested it on NanoHub. But a couple of people have finished the exercise. So let's ask one of them, so what do we need to put in here to make this work? Where do we start even to think about doing this?
I didn't complete, but I think the first thing is checking for the neighbors, right? If the neighbor is the medium, then you have a differentiation that should happen, right?
Yep. So we got to loop over all the cells, and we got to check. All right. So if we want to be able to do that, let's see. I guess we can skip through because we might not be able to do these codes. Let me go to the nucleate compartments 101 that has the solution in it. And so here's the solution, and we'll walk through these, right? So we're going to loop over. I usually like to do these with a Boolean flag or something like that I set up. So in contact with medium flag, it's set false to begin with. And I'm going to set some base probability to differentiate. And I chose 4 over 1,000 because that'll make the 1 over e time constant about 250 or 1 over that. And so within several time constants, it should fully differentiate if things are working. All right, so here, this for statement is where we're going to check to see whether the cell is in contact with medium. So
Okay.
I did the, yeah,
Sorry,
Jay,
Shane,
yes.
everything, nucleate compartments 1 and 2 seem to run fine on NanoHub. It's just the nucleate compartments with no extension. So
Okay,
if people will try loading the later versions, then see if those will work for you. Because I have no problem with the other ones. It's just that very first one, nucleate compartments, that has.
okay. Well, I'm glad these work at least. All right. So back to thinking about this here, right? So we're going to loop over this get cell neighbor data list again, and remember it's tuples. So the first element is the neighbor cell itself, a little bit better naming here. And the second element is the area, the contact area between cell and neighbor cell. If neighbor cell, so if the neighbor cell exists, we're actually passing. We don't want to do anything if there actually is a neighbor cell. We're testing whether it's in contact with medium. And this is where it's a little bit tricky, because if the medium is one of the cells that's in contact with it, then basically if medium will return as false. And so we do if neighbor cell, and if that's false, if that doesn't evaluate to true, then we end up under the else down here. And we can say, all right, the neighbor cell actually didn't exist when we checked it. That means it was medium. And so in contact with medium flag becomes true. And then if that flag is true and the cell type was mesenchymal, and a random number we pick is less than the probability, then we just set the cell type to epi. Questions on that implementation?
How could you write mesenc without the self?
Okay, good question. This is just, it's a programming hack that I like to do because it makes things a little bit easier for me when I read them. At the very top, I define some global constants that are just equal to the cell type numbers.
Oh, right.
And then I can use them throughout. And it just makes it more readable when I try to go through it.
Interesting.
what's happening.
Yes, interesting.
Great hack. I'm learning a lot.
And so if we run this one, then I hope this one will, I'm told it runs on NanoHub. I'll run it.
So
here
it looks
locally.
like I spoke too soon. It looks like 1 and 2 don't run, but 0 and 1, nothing and 0, 1 don't run. 2 and 3 run fine.
Okay. All right.
But
Well,
2, 3 and the full model run fine.
all right. Well, I'll run 1 here locally at least so people can see what its output should have been, where is the, let me pause this a second. Make this a little bit larger so we can see it. All right, it changed the color types on me, but that's okay.
I know what is the error. Actually, in nucleate compartments without any number, nucleate compartments CC3D, in line 25, cell list is typed in the old way, in the legacy, which is cell and then list with the uppercase L without the underline. And nowadays, actually, the right way to write is with an underline and L lowercase.
All right, so, oh, here,
Yes, this cell list should be underlined and then L
so,
lowercase. No,
re-
no. Cell underline list. Yes.
Underscore.
oh, cell
Underscore.
that makes sense.
Sorry.
Cell
Sorry,
underscore
sorry.
list. Ah, got it. Okay. All right, odd that some of them worked and others didn't then. That I would assume that was the same in all of them. All right. So if we go back and all right. So here's the run, right? In the first step, we've got all mesenchymal cells. They're in the sort of white there with yellow borders. And if we let this, I'll take a few steps through, right? It one by one differentiates cells on the border into this blue type epithelium. And they stick around once they differentiate. And I'll let this run forward. And it fills in, assuming we got the probabilities right. Right, we're a few hundred Monte Carlo steps. It's almost there. It'll finish this off. And there's our last one, 800 something, right? So it's stochastic, and there's a little bit of variability in when it will happen in each particular run, but that'll generate an epithelium on the outside. And if you killed off a few of the cells, it would generate more epithelial cells, right, to fill those in as it runs. Yeah. Questions? All right. And let me look at, I mentioned this before. Oh, sorry, back here to this one. If we really wanted to make it so that the probability, the rate at which things happen didn't depend on how often we called the steppable, we would uncomment out that line that has the 1 minus probability to the frequency, right? And then that would work appropriately. All right, so second exercise, we have a few minutes left. We can take that one on. So now we want to try to get something like this picture here, where we not only have epithelial cells on the outside, but they're polarized. They have apical and basal compartments. And so we want to write pseudocode that will differentiate cells on the outside to type epi, and then make sure that that epi cell polarizes and ends up with a basal and an apical compartment. And it's perfectly fine for the basal compartment to be the original epi cell type. We don't have to have a specific epi basal, but we do need a new apical domain. Right? I'll call it epi A in the one that we put together. And we want to generate that polarization, maintain it, and keep the polarized epithelium well organized so that the apical domains are on the outside in contact with medium and the basal domains are on the inside. Okay, so you can start with the solution that was posted for nucleate compartments 01 and nucleate compartments 02 will be where, well, it's where the solution is if you want to cheat and go ahead. But if you want to try to work on it yourself, I'll give you a couple minutes and then we'll look at the solution that's in that nucleate compartments 2. Nucleate compartments 1 does have the exercise written into it, so you can see it at the same time you're looking at the code.
Shane, I have a question. It's
Yeah.
more like a design choice and your experience with it. Have you, what is your experience of, instead of creating a new cell, making compartment, do you know if it makes different? I know that, yeah, of course, the whole model will work differently, right? You have to rethink the model if you're using 2 cells instead of making a compartment of a cell. But what is your take on the whole overview of this? Is there any benefits of on one instead of the other?
I like to have them as compartments in the same cell. And the reason I like to do that, because then you can link their behaviors together within a cluster. For example, suppose you have something that's dependent on the local concentration of a particular field, right? Then you could have a calculation of, all right, what's the concentration of the field at the apical domain? And then I use that to change the behavior of both the apical and the linked basal domain together. And that's just personal preference, the way I like to do it. I'm trying to think if there's any other specific reason for wanting to do that.
It definitely cuts some corners, especially when you're doing that kind of comparison, right? When you're using specific things for specific cells, then you don't have to assign to 2 cells. You can assign to a single cell.
Yeah.
A single type cell. That's what I mean.
A single type cell.
Yep.
Yeah. I mean, in some ways, it's a granularity of the model, right? Are you really trying to think about individual cells or just having an approximation of the different layers in the tissue?
Right. Yeah. Thank you so much.
Oh, you're welcome. You're welcome. All right, since we're coming up towards the end of the time we got together, I will go through quickly the solution for the one here. And again, we would have to change that to the new version. All right, so all this stuff about differentiating into epi is still there, exactly what was there before. But the new code block says, all right, if we have a cell of type epi, let's check whether it's appropriately polarized. So we'll set that Boolean false to start with, check over the compartment, and change that polarization flag to true if one of the other compartments happens to be type epi-A. Right? Just a little hint, if you're using CompuCell and you're using cluster cells and compartments, the compartment list that you get out of this get cluster cells, at least in the version that I was using, this may be updated, is not truly a Python list. So you can't iterate over it, but you can go from an I in the range of the length of it and access the individual parts of it. So it kind of behaves like a list, but not quite. It's not exactly iterable over. But assuming we do this, right, we find the polarization flag. If it's true, well, then great. It already has a complementary epi cell. If it's not, if the polarization flag is false, then we need to nucleate a new one. So we create the new epi-A cell. We get the contact, we get the list of pixels in contact with medium and in contact with other kind of surfaces. And in this case, we're going to assign some of the pixels in contact with medium to the new cell. We want the apical cell to differentiate on the outside. Then we're going to set its target volume, lambda volume, target surface, lambda surface. And lastly, make sure its cluster ID gets set to be the same as the basal domain from which it came. So if we run this one, you can see the epithelial cells pop up there, and as they do so, we get epi cells that come up on top of them as well. And occasionally there'll be one like here at the top that takes a long time to get an apical domain. That's because of the way I wrote the probabilities here. If it doesn't have quite enough contact area, then the probability is pretty low. Eventually there'll be a fluctuation where that one probably gets one, but it's having a hard time getting the random number to let that one get an apical domain. Okay. But by all means, you have all the exercises there and the solutions for them. If you work through them and you have questions, feel free to ask. Happy to think about this as a conversation on how we do these kind of models and how we develop them.
I have a question if we still have time.
Okay.
Talking now more about turning this model into a truly useful model. I saw that you had many parameters. You had many fields, many types of cells, and clusters, and links, and some cells with high lambda volume, some, many types of mechanisms. How did you fine tune everything?
That's a great question. So how do you tune the parameters? How do you figure out what works? I often felt like I was in the middle of like this optimization problem, and it was really just me using intuition to say which region should we explore and which direction should we go. And a lot of it was sort of working backwards to say, all right, we want this behavior. Here's how we could completely script that behavior, which feels like cheating in the model, right? We're not really letting the regulatory elements generate the behavior. And then once you understand, well, this is what it would take to script it. All right, how do we think about the interaction of the different fields and contact and neighbor behaviors that would give us that same result and would give it to us robustly? It really was sort of building it one step at a time and one behavior at a time and think about how do we get this. I mean, it was a good year's worth of work where I basically got to act like a postdoc and focus on this for a year. So.
Oh, so the point that I'm interested here. So you took every mechanism, you treated them individually, or you treated them in composing with the previous mechanisms? Like you have the model, then you add something and study how it works with everything you already have, or you study that mechanism in another simulation isolated and then add both after?
I did a little bit of both. There were certainly times where I would make a much simpler similar simulation just to test a mechanism and say, how would I make this work? How can you code it up? And then put it into the existing larger model.
And you didn't use any type of optimization?
No, I can't say that there was. I mean, it really was. I think the optimization was in terms of thinking about the structure of the model. The test for the structure of the model was could it get the in silico knockouts right? And then in terms of the parameters of the model, think about, are they even in the right ballpark? Was the kinetics of growth and fusion on the right time scale? Right, with some assumption that I have to make about how Monte Carlo steps turn into days. And that was basically on convenience, right? How long a model do I actually want to run? If I want to simulate 3 days, I got to make sure the Monte Carlo, it doesn't take all that many Monte Carlo steps per day.
Yes.
But I think, like the Poisson probability trick, I think having things like that are really important for optimizing the model, because you can test lots of parameters with a long time between stepables, right? A large stepable frequency, only calling it every 100 or 500. And then once you establish, oh, okay, this is the right parameter range based on that, you can go back and call the stepable more often to get more realistic behaviors, but you don't have to test it at that level because it's the stepable that's the slow part.
Oh, so I didn't catch that part, I think. So you're saying that you introduce a mechanism, you make it really strong, but called less frequently?
That's the way I generally did it, because the models would run much faster if I called the separable less frequently.
And you just make the effect stronger.
Yep.
I didn't know. I never saw this. It's a good idea.
Another thing which we don't advertise is that if you change the flip to dim ratio, which is a flag we almost never use in the XML, you can speed up the code a lot. Basically, it increases the speed at which certain elements are called compared to the POC step, reduces the number of times you go through the Python layer. I didn't expect that would have a big effect on overall code execution, but it does. So are there other questions? We're going to have to stop in just a few minutes.
I have a quick
wanted
question.
to introduce themselves
Yeah.
to Shane and ask a question or two. Please go ahead, Norma and Jasmine, please go ahead.
Oh, my name is Jasmine, and I'm a student at Morehouse College. And I had a question about, I guess, like how, because I don't really have much experience for coding. So how did you come up, were able to make like a code to connect all the different the different pathways you show? Because I'm also interested in connecting more than one pathway to the model.
Yeah, I mean, it's a very incremental process, right? And it helps to think about modules that are within your pathways, right? That's why I emphasize like that auto activate, that mutual activation loop and that mutual inhibition loop. So I would grow sort of the complexity of the model by saying, all right, I know this loop is central to it. So let me make sure I get it right where, you know, Sonic and FTF10 are behaving appropriately and giving the right kind of behaviors. And once I have that, then I can add on these accessories that fine tune it, right? FTF7 or BMPs, right? That give me a little bit more tuning in it, but I want to make sure I get the central element of it correct first. And it really is being very systematic and taking a lot of notes. I wrote a lot of notes in my notebooks as I went through developing the models about what works and what doesn't, but thinking about how do I gradually add on to it, right? And build the model up one additional complication at a time.
Thank you.
You're welcome, Jasmine.
I have a question in line with that. My name is Nasheen. I'm a PhD student at SUNY Binghamton. So I'm also trying to do multiple signaling pathways, trying to integrate them into CC3D. So I don't know, like, I have started writing my different pathways in antimony, and then I'm thinking of like integrating them into CC3D. Do you think that's a good approach?
I think it's a good approach. It's not the one I took, right? I don't actually, the model that I presented here doesn't have any cell internal signaling. It's
Okay.
all based on just what secreted signals are present at what other cells. And it's directly
I saw that.
behavior to those concentrations and not having the intracellular. The intracellular would be nice, and that's a really nice addition that antimony brings in. I just wasn't ready to do that when I wrote this particular model.
And can I contact you later on if that's a possibility, if I have any more questions?
Certainly.
Thank
Oh,
you.
wait a second.
Thank you for
My email address here is incorrect.
Oh, okay.
There's no M dot in front of it. It's just shane.hutson@vanderbilt.edu.
Oh, okay. Thank you.
Oh, thank you.
There's a problem about this notion, and Shane, is that I think Tellurium wouldn't work. I don't know how Tellurium would improve Shane's model, because Tellurium doesn't offer you doesn't offer you a way to solve the equations through space, and Shane needed the fields to diffuse, right?
Yeah, indeed. It was the spatial distribution of the fields that was really important, and their overlap.
And Tellurium doesn't solve this. Tellurium only solve the as mean field equations, the chemical reactions.
Well, I mean, you could you could do it, although I suspect it would be pretty slow if you had tellurium running basically to solve the mean field within each cell. That would be enough.
Yes.
You don't have to have the gradients over the cell, but you would have to have it running independently in every cell.
Yes. And you have also to, and this is I don't know how to do this, but you would need to transfer field from cell to cell to model
Right,
the
well,
diffusion part.
Right, well, we do that. We
Okay.
do that quite a bit, actually. So I think it's possible. You're raising a very profound issue, which is when do you need to have a diffusion field, and when do you need to have, when is it better to represent concentrations inside of a cell as a scalar? And they both have advantages and disadvantages, and coupling a scalar inside a cell to a diffusing field outside the cell is certainly something that you could do. And I don't think I think they're different points of view. They may have different efficiencies. But I don't think either one is completely right or wrong, because they're both simplifications of a more complex real situation.
Yeah, I was just thinking with regards to what makes more biological sense. And I might be wrong. I don't have like a lot of modeling experience, so
No, that's a classic trade-off, is fidelity to the biology versus actual feasibility of the modeling, right? And where do you fall along that continuum?
Are there any final questions for our speaker? We want to thank Shane. That was a great presentation. I would love to see this, the full model, both of these things put up as NanoHub demos, because I think they're both tremendously educational.
Okay, we'll
would
work
be
on that.
great.
And congratulations for the model. It is a very, very impressive model. I think you got in the right point between complexity and simplicity. I think you got in the balance between biology and computation here.
Well, thank you.
Very nice job.
Thank you.
Okay, well, people are free to discuss. We'll probably start again we'll probably start a little late as usual. We were a little late getting started here. Julio isn't back, so until Julio is back, we can't go on to the next module in any case. So please feel free to have a conversation. If you want to take a break and have a cup of coffee, that's fine too. And I'll see you back here probably at 4:40 for the next module. But you certainly are free to stick around and chat now.